Welcome back to our second lesson on genetics. We're going to continue on today to learn more about what Mendel did as he was studying his pea plants. And remember, everything Mendel found applies to both plants and animals. So today we're going to learn about the law of segregation. And we're also going to talk about making predictions, how we can predict what possible offspring two parents can have. In the P, remember P stood for parental generation. The F stands for filial, and then you can have first, second, third, and so on, depending on how many generations you are following. So F for filial. So in his pea generation, Mendel had a green pea pod plant, and he crossed it with a yellow pea pod plant. And if you notice, all of their offspring were green in this F1 generation. So that means, which trait do you predict was dominant? It is the green trait. So that means green must be dominant to yellow in terms of pea pod shape. So Mendel always chose purebred plants for his pea generation. They were pure, or also known as homozygous. So that means big G, homozygous dominant, and little g, homozygous recessive for our yellow plant. The only possible eggs or sperm that this plant could make are ones that would have a big G allele. And for this one, the only possible egg or sperm it could make, pollen, would be ones with the little G allele. Remember, these, even though we're not drawing them, they represent alleles found on homologous chromosomes. And what happened to homologous chromosomes during meiosis? What happened to these during anaphase 1? They were separated, and only one goes into each egg or sperm cell. So again, we're drawing it like this, but these are still alleles on homologous chromosomes, one from their mom parent, one from their dad parent. And homologous chromosomes, they have more than one gene located on them, but we're focusing on just one at a time so far. So just one gene at a time. So Mendel had another question. Had the recessive alleles disappeared, or were they still present in the F1 generation? So here in the F1 generation, he was like, what happened to this recessive? What happened to the yellow? Why are all their children green? And so in order to figure out if this yellow allele, this recessive allele, had disappeared, he bred two of the F1 plants and then observed their offspring in the F2 generation. So he allowed them to self-pollinate, then he planted their seeds, and he discovered that the recessive trait reappeared. How did that happen? And so if you remember, the only way to get a recessive phenotype is to have the recessive genotype. And we're going to learn how to figure out the rest of these. So this is referred to as the principle or law of segregation. It's the separation of alleles during meiosis. Remember, alleles are found on homologous chromosomes. So they're drawn here, but you could also draw them like this. They're on homologous chromosomes, and they are separated in meiosis. So each egg or sperm only gets one copy. So alleles for each trait. So we're only looking at one trait here, let's say for height or tall plants or short plants. But for every gene, even color, shape, size, flowers, every gene is separated during meiosis. So they segregate or separate. It's also referred to as the law of segregation, but you can think of separation. We're separating these alleles on homologous chromosomes. And again, we're separating the alleles for one gene, trait, or characteristic, and they're going into gametes. Each gamete, this is really important, only gets one copy. Each gamete only gets one copy, because remember, gametes are haploid. One copy or one set of chromosomes, because it is haploid. Each individual gets one allele from mom and one allele from dad. So you get one homologous, or one set of chromosomes from your mom, one set of chromosomes from your dad. Same with plants. So gametes are haploid. They only have one allele for every trait. Really important. So here we're only looking at one trait, so that's why I only see one allele in this gamete, but it would actually have an allele for every trait. So if we looked at flower color, big P, those would also be separated too. So again, this goes for every trait, only one allele. Green and yellow. So again, for every trait, you'll only find one allele in each gamete. So again, law of segregation, if this parent, so that's what we saw in the F1 generation, they were all heterozygous because they had, in the P generation before them, the parents were big G, little g, little g. If we cross these two plants, the only egg or sperm that this plant could make would be gametes with G's. They would all be the same. Only G's. And if we looked at the gametes after meiosis for this plant, they would only have little g's because of the law of segregation. And so if we fertilize these two plants, that means their offspring would be, when these come together, big G, little g. So here's our F1 generation. And they're green because green's dominant over yellow. But then if we cross two of these plants, now this F1 generation, their egg or sperm could either have a big G dominant or a little g recessive, and that goes for both of them. And again, there's 4, so it'd be 2 and 2 for each of those alleles. The offspring then could be any combination of these 4 egg and sperm cells. We don't know which one is going to come together. So again, when we are talking about genetics, it's always a probability. We don't know which ones are going to come together. It is a prediction. And they're not always going to have just 4 offspring. There could be more or less offspring produced each time. For humans, you only get, most of the time, 1 offspring at a time for fertilization and reproduction. Sometimes twins, triplets, but those tend to be rare. For dogs or cats or some plants, you can get 9, 8, 6, or a lot more. So prediction. And we're going to be talking about ratios and percentages. So the law of segregation is just talking about 2 alleles. For one trait, they always get separated into different egg and sperm cells. Each egg and sperm can only have 1 allele for every single trait. That's what Mendel figured out because he saw the yellow again in the F2 generation. Going into some probability. Again, we're talking about probability and heredity, the study of genetics. And we use probability and statistics a lot for this. So it's the likelihood that a particular event will occur. And again, we're using probability because we don't know how many offspring are going to be at a time, and it's just a prediction for what offspring they could have. So if you flipped a coin, then you would see that there's only 2 possible outcomes, heads or tails. The probability that a single coin flip lands heads up, there's a 1 out of 2 chances, or 1/2 or 50%. But what would you predict the probability of flipping a coin heads 3 times in a row? So to get heads on a penny, you have a 1/2 chance. To get that 3 times in a row, these events are independent of each other. Flipping a heads once doesn't necessarily mean that you're going to flip it again. They're not correlated. So to figure out 3 times in a row, you do 1/2 times 1/2 times 1/2, because there's a 50% chance for each event, and they are separate events, so the total would be 1/8 chance of getting 3 in a row. What if we said getting 5 tails in a row? And you had guessed it. Again, if you said multiply 1/2 by itself 5 times, that is correct. So 1/2 to the 5th power instead, and so forth. And this goes with genetics as well. Every organism has 2 alleles for 1 gene. So there's a 50% chance of what allele you're going to inherit from that parent. Law of segregation. So probability and alleles. So Mendel consistently saw the same ratio in the F2 generation. So F1, they were both heterozygous when he let them cross or self-pollinate. And he saw in the F2 generation that there was about a 25% chance, or 1/4, of getting a homozygous dominant. There was a 50% chance of getting heterozygous, 2 out of 4. And again, this is just prediction. And there was, again, a 25% chance of getting a homozygous recessive or yellow offspring. And these ratios are exactly the same no matter what trait you're looking at for any organism. If both parents are heterozygous, you will always see this predicted ratio, and the more offspring they have, the more closer to this predicted you will get, but the numbers will be the same ratio. Again, the more offspring they have. The ratios we then look at are genotype and phenotype ratios. So if we look at genotype, the genes, that's the ratios we just wrote down. We always start with homozygous dominant. So genotype ratio. Homozygous dominant is written first. And you can write it as a percent or a fraction. And so ratios, then you would write then heterozygous, which is 50%, to our homozygous recessive. That's our genotype ratio. And Mendel always saw that after crossing 2 heterozygous parents. What we're also going to learn when we do predictions is we're also going to write a ratio for phenotype. Physical. And so phenotype ratio, you first write the dominant trait, so that would be green in this example. The dominant trait comes first. And there are going to be 3 out of these 4 that could be green. And again, this is a prediction. And you could write it as 75% as well because it is a prediction. We don't know if they're going to have 3 offspring. Every time, could be more, could be less. And 25% recessive trait. And he saw these ratios almost every time, and we'll see these in our examples. So monohybrid cross. So I want you to copy down this example. Some of you may have done this before, and that's great. But this is a tool. These Punnett squares are tools to help us make predictions for possible offspring that parents could have, or the organisms that we're starting with, our P generation. We are going to first just look at one trait at a time. This is called a monohybrid cross, one trait or gene at a time. But we will also later in this unit be looking at multiple genes at a time, more than just two alleles, because we're going to look at multiple genes. So monohybrid cross, it's to predict the possible phenotypes and genotypes of offspring. Monohybrid is just one trait at a time. And again, these are predictions. So the way a Punnett square works, you write the possible gametes for the traits of the parents on the axes. So the axes represent gametes of parent generation for trait. And the boxes on the inside represent possible offspring. And it's kind of like a grid. So as you see with the arrows here, this letter gets carried all the way down, this one gets carried all the way down, this carries across. The dominant letter is always written first, if it is there. And then we write the recessive. So here in this example, the big R would get carried all the way down, so big R, and then this little r would get carried down. I'm going to write it here just to be safe because recessive gets written second. So little r, and then we'll move on to here. This one gets carried across. So another big R, and down here as well, and across here, big R, little r, little r, little r. So here we're talking about round versus wrinkled, so the seed shape. Round seeds are dominant to wrinkled. So for this offspring, or a cross between these two parents, their possible offspring, they could have one homozygous dominant, so 25%, or you can write it as 1 or 1/4. That is up to you. Remember our ratio, we write homozygous dominant first, then we write that possible heterozygous offspring. Here we have two possible heterozygous offspring. So you could write 2, 1, 2, 1/2, or 50%. And then we only have one possible homozygous recessive or wrinkled offspring. So again, that would be 25%. For our genotype ratio, so we're making predictions. And for our phenotype ratio, we would have 3 round, that's dominant, so you can write it as 3 or 75%, and only 1 wrinkled, 1 recessive. 25% or 1/4. So practice. What phenotypes would you expect in the F1 generation? So we have a parent that is homozygous dominant. So what gametes, if the alleles segregate in meiosis, that means they could have gametes with a big R or a big R. So we'll write these here. Those are their only possible gametes. And for this parent, if their alleles segregate, they're only going to have offspring with little r. So those are their gametes. So what are their possible offspring? So a cross between two homozygous individuals would result in what phenotypes? Would their offspring be round or wrinkled? And since all of these genotypes are the same, that means the phenotypes are all going to be the same. So they have 100% round offspring, and the reason that we say round is because it asked for phenotype and not genotype in this example. Example number 2. A homozygous dominant and round seed plant is crossed with a homozygous recessive wrinkled seeded plant. What are the genotypes of the parents? For this one, we don't have to do a cross. It's just asking the genotypes of the parents. So homozygous means 2 of the same alleles, dominant and round. So big R, this is parent number 1, is crossed with a homozygous recessive wrinkled. So parent number 2 would be little r, and that's the genotypes of the parents. Example 3. In Labrador retrievers, the allele for black coat color is dominant over the allele for brown coat color. If a brown Labrador retriever, so if it's brown and brown is recessive, that means this parent has to be little b, is crossed with a heterozygous black Lab retriever. Heterozygous, two different alleles. So big B, little b. Which of the following genotypic ratios could be expected? So it shouldn't be which of the following, not multiple choice. Which could be expected? We'll do this cross. So draw your Punnett square on your paper. And so it does not matter which side you put which parent. The outcome will be the same. So for this parent, the only possible gametes would have the alleles for little b and little b. For this parent, their gametes could be a big B or a little b. So those are their gametes. Remember, parent can only pass on one allele for the trait because the other parent gives it to the offspring as well. So big B, little b, little b, little b, big B. So what is their genotypic ratio? Genotypic ratio. So for genotypic ratio, we write homozygous dominant first, and there are none, so 0 homozygous dominants. They cross between these two individuals will not produce any homozygous dominant offspring. So next we write heterozygous, and that is 50% or 2 out of 4. You can write it however you'd like. I'll change it up and do it this way this time. And homozygous recessive, there are 2 possible outcomes. So this is our genotypic ratio for this example. Purple is dominant to white. A heterozygous purple flower is crossed with a white flower. So heterozygous purple flower is crossed with a white flower, which is recessive, so little p, little p. What are the genotypes and phenotypes of their offspring? So again, draw a little Punnett square on your paper and fill in their possible gametes of the parents and cross them. Let's see what the predicted offspring could be. So again, the Punnett square is just a prediction. It doesn't mean they're going to have 4 children. So for genotypes, it doesn't ask for a ratio. It asks what are they. So they could have big P, or little p, little p. And that would be 50-50 if you were doing a ratio. And for phenotypes, they could have offspring that are purple because the heterozygous, or they could have offspring that are white because of the homozygous recessive. Last example. Long tongues are dominant to short tongues in tree frogs. What is the probability that two frogs that are heterozygous for long tongues could produce offspring with short tongues? So what is the probability? We have two heterozygous frogs. What is the probability that their offspring would have a short tongue? So set up your cross. So if it has a dominant allele, it's going to show the dominant trait. So that means these three will have long tongues. So there's a 25% chance that their offspring would have short tongues. All right, it's time for you to practice on your own.